After studying this module, you shall be able to learn about reactive organic species and the potential toxicity exhibited by them, understand why nature has evolved efficient protein complexes called catalases and peroxidases to catalyze the decomposition of one of such frequently encountered ROS H2O2, know the structure and function and mode of action of catalases, which is one of the most vital enzymes found extensively in all living organisms exposed to oxygen. Know the structure, type and mode of action of peroxidases, horseradish peroxidases, which are heme containing proteins found from root extracts of horseradish plant that act as catalysts to promote the oxidation of various compounds by reducing peroxides. The ability of organisms to utilize molecular oxygen for performing diverse biological functions has proven to be a major evolutionary breakthrough. However, this evolution has come at a cost. Even though essential for almost every form of life on earth, oxygen has unfortunately been the source of various toxic substances such as hydrogen peroxide and superoxide, often termed as reactive oxygen species or ROS. Now, what is hydrogen peroxide? Hydrogen peroxide is a frequently encountered ROS with the chemical formula H2O2, is a byproduct of various reactions in organisms. It is generated as a consequence of oxidative cellular metabolism, which may eventually get converted into highly reactive hydroxyl radicals as shown in figure. These radicals may then cause damage to a variety of cells, leading to oxidative stress and ultimately death. Considering the potential toxicity associated with this ROS, its rapid and efficient removal is of utmost significance. Fortunately, nature has evolved efficient protein complexes called catalases and peroxidases that are able to catalyze the decomposition of this reactive molecule H2O2 at reasonable rates. Scheme depicting the generation of harmful ROS and the consequent damage caused by them. So, let us begin with catalases. So, what are catalases? Catalase is one of the most potent heme containing enzymes that catalyzes the breakdown of hydrogen peroxide into water and oxygen as demonstrated in figure. This enzyme remained unnoticed until 1818 when Louis Jacques Thénard, the discoverer of H2O2, suggested its decomposition caused by an unknown substance. Found extensively in all living organisms that are exposed to oxygen inside the cytoplasm of the cells, this enzyme protects the cellular organelles and tissues from the harmful effects of H2O2, which is continuously generated by numerous metabolic reactions without the formation of free radicals. Amongst a myriad of enzymes, catalase has one of the highest turnover numbers, as each catalase molecule possesses the capability of decomposing millions of hydrogen peroxide molecules every second. The reaction of catalase in the decomposition of hydrogen peroxide in living tissue is demonstrated in the following equation. As you can see, two molecules of H2O2 break down into two molecules of H2O plus O2. Figure represents the decomposition of hydrogen peroxide by the enzyme catalase. Now, what is the structure of catalase? How does it look like? Most catalases exist as tetramers of 4 polypeptide chains, each polypeptide chain consisting of approximately 500 amino acids. Every monomer unit is further comprised of an active heme unit, that is iron 3 protoporphyrin prosthetic group, that is embedded deep within the structure at the catalytic center. The crystal structure of Helicobacter pyrrole catalase has been depicted in figure through a funnel shaped channel which is approximately 30 angstrom long and 15 angstrom wide. Further, it is lined with hydrophilic residues at the entrance and hydrophobic residues as the channel descends. The iron 3 of the heme unit exists in high spin state and is coordinated to 4 nitrogen atoms of the porphyrin ring, while the 5th and 6th axial positions are occupied by phenolate oxygen of the tyrosyl residue and water molecule respectively. During catalytic reactions, the axial water molecule is displaced by hydrogen peroxide. It is the structural rigidity of catalysis that imparts it considerable stability, making it resistant to pH, thermal changes, unfolding and denaturation. So, figure represents 4 subunits of Helicobacter pyrrole catalase colored in blue, red, yellow and green, heme binding pocket of one of the subunits blue and molecular structure of heme. Next, we move on to the mode of action, that is, how does catalase work? Although the exact mechanism of catalase is not known, yet it is believed to occur via a two-stage mechanism in which the heme iron is alternately oxidized and reduced by hydrogen peroxide at the active site. The first step involves the oxidation of heme to an oxyferryl species by one H2O2 molecule. So, here iron is changing from +3 state to a +4 state, while the second step involves the regeneration of the enzyme by another hydrogen peroxide molecule to produce water and oxygen. So, we shall go into the details of the molecular mechanism and structural changes involved. What happens actually is that during the catalytic reactions, hydrogen peroxide interacts with catalase by displacing a water molecule and coordinating through one of the oxygen atoms. The hydrogen atom bound to the coordinated oxygen gets hydrogen bonded to the nitrogen atom of the imidazole N of the histidinyl residue of the protein chain, thus developing a delta positive charge on the coordinated oxygen. The other oxygen atom develops delta negative, which is further accentuated by electrostatic interaction with positive charge of the arginyl residue of the protein. Thus, there is a charge separation between the two peroxo oxygens. This leads to heterolytic fission into neutral oxygen and O2 minus. The latter combines with 2H plus forming water molecule, while the neutral oxygen takes 2 electrons forming O2 minus. 1 electron comes from iron 3 forming iron 4 and second electron from porphyrinate ring forming porphyrinate -1 ring, which is a pi radical. This intermediate radical may be stabilized by the transfer of negative charge from the protein side chain due to the pi contact or hydrogen bonding. Thus, the oxygen of the oxocation is closely surrounded by the protein. This is called compound 1. It has been shown in figure where we can see how the compound 1 is getting formed. In catalase, one molecule of H2O2 takes part in the formation of compound. The O2 2 minus of the second H2O2 molecule loses 2 electrons in steps to form compound forming oxygen and 2H plus combined with O2 minus of the compound to form water molecule as shown in figure. It is worth mentioning that both the oxygen atoms of the oxygen molecule come from second molecule of H2O2, and this has been confirmed by using labeled H218 isotope of oxygen. So, figure shows the mechanism which illustrates how the catalase enzyme precisely works. After having a thorough detail about what catalases are, we shall next move to peroxidases. So, what are peroxidases? Peroxidases are heme containing proteins often found in plants and animals including humans. They act as catalyst to promote the oxidation of various compounds by reducing peroxides, which is one of the toxins produced during various biochemical reactions. Especially, it breaks down hydrogen peroxide to form water, which can cause damage due to its oxidizing properties. The peroxidase reaction is shown as follow. You can clearly see how one molecule of H2O2 is reacting with one molecule of AH2 to form two molecules of water and plus A, wherein the compound AH2 is getting oxidized to A. Although there are different categories of heme peroxidases, extensive research has been conducted in literature to understand the structure and function of horseradish peroxidase, which will be discussed in the ensuing sections. So, what is a horseradish peroxidase? Horseradish peroxidase, an ubiquitous oxidative heme containing enzyme derived from the root extracts of horseradish plant, utilizes hydrogen peroxide to oxidize a wide variety of inorganic as well as organic compounds. Horseradish peroxidase is brightly colored, especially under the near UV light, and hence can be attached to transparent proteins so that they can be seen under different wavelengths. It should be noted that when it is combined with other compounds, it exhibits high reactivity towards human tumor cells. Further, there are several distinctive peroxidase isoenzymes known, amongst which C-isoenzyme is the most abundant. Let us see how the structure of horseradish peroxidase looks like. 1 of iron heme group and 2 calcium atoms, which are essential for the structural and functional integrity of the enzyme. The heme group has a planar structure with the iron atom in the middle of the porphyrin ring having 2 open bonding sites. The heme group is attached to histidine at proximal histidine residue, which is histidine 170, by a coordinate bond between the histidine side chain nitrogen atom and the iron atom. The second axial coordination site, often called as distal side of the heme plane, is vacant in the resting state and becomes available for hydrogen peroxide to attach during reduction oxidation reactions. During this activation process, the bonding of hydrogen peroxide to the iron atom generates an octahedral coordination around the iron atom. To make this structure more clear, the 3-dimensional view of horseradish peroxidase has also been provided. As shown in figure, the iron heme is located at the center of the enzyme, represented in black, while the iron atom as a red sphere. The 2 calcium atoms, on the other hand, are shown as black spheres, are located within the helical regions of the enzyme at positions distal and proximal to the heme plane. The alpha helical and beta sheet regions have been represented as the multicolored helical structures. Each calcium site of the enzyme is 7 coordinate with oxygen donor ligands provided by a combination of amino acid side chains, carboxylate, hydroxyl groups, backbone carbonyls, and a structural water molecule. It should be worth noted here that both the heme group and the calcium atoms play vital role in the proper functioning of the enzyme, and the loss of one would result in instability. Figure 6 shows the structure of iron heme group in horseradish peroxidase. There is a histidine enzyme attached to the proximal histidine residue under the heme group plane. Figure shows the 3-dimensional view of horseradish peroxidase. While the heme group is located at the center and is mostly black with the iron atom in red, the calcium atoms are black spheres and the alpha helical and beta sheets are surrounding the heme group. Next, we move on to the mode of action, that is, how does the peroxidase enzyme work? The first step of the redox reaction of horseradish peroxidase is the reaction between hydrogen peroxide, that bonds to the vacant octahedral position on the iron atom as shown in figure. So, you can see iron 3 enzyme is getting converted to compound 1, which is a pi cation radical when hydrogen peroxide interacts with it, removing a water molecule similar to the case of the catalase enzyme. In the next step, iron 4 compound 1, which is a pi cation radical, forms compound 2, where it accepts the compound 1 accepts 1 electron and 1 H plus to form compound 2. In the next step again, compound 2 accepts 1 electron and 1 H plus to form a water molecule, while the iron 3 enzyme is regenerated back. What happens if compound 2 interacts again with a hydrogen peroxide molecule? If it does so, compound 3 is generated as water molecule gets eliminated and iron 3 gets coordinated to OOH. In the next step, the OOH radical is eliminated and iron 3 enzyme is regenerated back. So, thus we can see during the course of reaction, 3 intermediate horseradish peroxidase compounds are formed as shown in figure. They are generated with either an addition of an electron or a reaction with hydrogen peroxide. The reduction of compound 1 to compound 2 and compound 2 back to the rest state is done by reduction substrates, namely phenols or aromatic amines. Let us now talk about the differences of catalases and peroxidases. You know, while catalase catalyzes the breakdown of hydrogen peroxide into water and oxygen, peroxidases on the other hand promotes the oxidation of various compounds by reducing peroxides. The second difference between catalase and peroxidases is that while catalase compound 1 oxidizes a second hydrogen peroxide molecule to oxygen, peroxidases uses compound 1 to oxidize other substrates rather than hydrogen peroxide. Besides this, there are also the structural differences between catalases and peroxidases, which we have described earlier. So, thus recapitulating the entire lesson, we can summarize, although essential for almost every form of life on earth, molecular oxygen has unfortunately been the source of various toxic substances such as hydrogen peroxide and superoxide, which are termed as reactive organic species or ROS. Hydrogen peroxide, which is one of the most frequently encountered ROS, is a byproduct of various reactions in organisms and generated as a consequence of oxidative cellular metabolism, which may eventually get converted into highly reactive hydroxyl radicals that eventually cause damage to a variety of cells, leading to oxidative stress and ultimately death. Therefore, nature has evolved efficient protein complexes called catalases and peroxidases that are able to catalyze the decomposition of hydrogen peroxide at reasonable rates. Catalase is one of the most vital enzymes found extensively in almost all living organisms exposed to oxygen that catalyzes the breakdown of hydrogen peroxide into water and oxygen. It exists in the forms of tetramers containing 4 polypeptide chains. Every monomer unit is further comprised of an active heme unit, that is iron 3 protoporphyrin prosthetic group, that is embedded deep within the structure. Even though the exact mechanism of catalase is not known, yet it is believed to occur via a 2-stage mechanism in which iron is alternatively oxidized and reduced by hydrogen peroxide at the active site. Peroxidases are heme containing proteins found in plants and animals, including humans, and act as catalyst to promote the oxidation of various compounds by reducing peroxides. Horseradish peroxidases are ubiquitous oxidative heme containing enzymes derived from the root extracts of the horseradish plant that utilize hydrogen peroxide to oxidize a wide variety of inorganic as well as organic compounds. The major difference between catalase and peroxidase is that catalase produces water and oxygen, while peroxidase generates water and an activated donor molecule.